it was found that the way rapamycin works is that it actually forms a complex with two other proteins. The first protein is FKBP12. This is that's short name for FK506 binding protein, and it's number 12 because now there are dozens of these that have been identified. So it actually binds this FKBP protein in the cell by virtue of this binding motif. It then, that complex, but not rapamycin itself, but that complex then binds this protein, which is mTOR, target of rapamycin, right? It was discovered because rapamycin bound to it, but rapamycin only when it's bound to FKBP12. So it turns out that rapamycin recruits FKBP12, or vice versa, right, and binds mTOR. Well, mTOR turns out to be a central kinase in metabolic control of cellular proliferation. So, and it turns out that the mTOR is a very distinct target for T cell proliferation. So what this does is it recruits mTOR away from its normal function, and it essentially shuts down its mTOR function. mTOR binds to other proteins normally, but this complex is so tight that the mTOR is virtually locked down and cannot operate. So you basically shut down T cell proliferation. So as you can see, this mechanism would never have been found by designing. The smartest people around could not envision this mechanism at the time or even now, I don't think. So for this to have been found, it was found in a phenotypic screen. And now there are several lines of mTOR inhibitors. There are synthetic molecules that are now reaching the clinic for cancer and immune suppression. So again, it's an example of a privileged structure. So what is interesting now is that this illustrates the concept of motifs that bind proteins in natural products that you can re-engineer to bind new proteins. So we know that rapamycin in this case binds FKBP12 by virtue of this motif here. And this is the motif that binds mTOR, right? So we knew that certain members of this immunophilin class show neuroprotective properties. But it was also clear that rapamycin, this part of the molecule, was not necessary for that. So actually, it turns out that if you take this triene here and you do a simple Diels-Alder addition to the right-hand dyne of this and begin to append modifications, this changes the conformation of the rapamycin ring quite dramatically. It no longer binds mTOR, so it's not immunosuppressive anymore. But what it does do is, first of all, it actually stimulates neural outgrowth. And this is all published in this paper in PNAS a couple of years ago. You can look up the details. But it turns out that it stimulates neural outgrowth, and it also provides very strong neural protection. And you can see the dose. It's a very, very straightforward dose-response phenomenon. Turns out it does this, and we're not sure how this all works yet, but it actually doesn't bind mTOR, but it binds the beta subunit of the L-type calcium channel. It also binds a different FKBP. It binds FKBP52. So somehow these three proteins are now related. Turns out that this molecule has very good activity in animal stroke models by virtue of this neurite outgrowth and neuroprotection activity. So we're very encouraged by that. And all of that is published in this paper, the animal model data as well as the in vitro data and some of the chemistry. So finally, the final example is something a lot simpler. This is nicotine. We saw this molecule before. Very simple molecule, a little pyridine ring, and then a parole here, a tetrahydroparole actually. And so I don't have time to get into the pharmacology of smoking. It's fascinating, and I don't know very much about it anyway, so it's convenient that I don't have any time left. But there's a molecule here that has been known for some time to have similar nicotinic activities as nicotine itself. This is cytosine. It's made by this plant here. I forgot. Oh yeah, lupinus. So this is actually a very large family of plants, but it has very poor drug-like properties and really wasn't something that could be advanced. But these organisms here actually took this molecule and then synthesized analogs to impart the right properties. And now this is actually I'm going to call it Shantix because I can never pronounce varenicline. So you can see how the structure of this, actually we could draw nicotine a little bit differently and draw it so that the pharmacophore would be positioned the same way versus the pyridine. You can see actually how this now is pretty much incorporated. The reason this molecule was made was that it was hypothesized that a partial agonist of the nicotine receptor would be an effective smoking cessation drug. So actually Shantix is very successful. I think you can see it has a very strong clinical success rate. And this is an example of a much smaller molecule that you can, from plants, that you can take forward. So in summary, natural products have been and continue to be a rich source of chemotherapeutics. I think there's no doubt about that. They are biologically privileged chemical structures with which to attack previously undruggable targets. I think we saw examples of that. They are biologically privileged structures which can be leveraged for new biology and new targets. I think we saw an example of that. And complex natural products can access desired drug property space. I think we saw some examples of that. So thank you for your attention.
Okay, we're on time, and this is the end of the session. So speakers can come up while we're having specific questions for this presentation. That would be great. Entertain any roundtable discussion.
Hey, Marty, I want to thank you for a great session, to all the speakers. I just want to make a comment and then I have a question. I think one of the things that impresses me here is that we think of basic research as being research and this being sort of applied science, but I think you can see that drug discovery is very much research, and it's very creative and very exciting, and you don't really know which way you're going all the time. I think somebody mentioned earlier that the ultimate action, I think it might have been Laura, that the ultimate action may not be related to the initial mechanism of action, and you're just kind of being the ultimate observer as you go through the drug discovery process. So I think it's a great example of that. My question is two questions about natural products. One is when you mentioned the diversity, the chemical space and the infinite nature of protein sequence in terms of all these other things, including folding, you didn't mention anything about post-translational modification, which also adds tremendous complexity, especially in animals, let's say. And I just wondered, my first question really is, what about post-translational modification in natural products?
Well, there is post. You talk about the small molecules themselves, the natural products, or the proteins that they bind.
Well, actually, I guess I'm talking really about the small molecules that these plants and whatever are making.
Yeah.
Are they post-translationally modifying them?
Yeah, they are actually. They're the core of if you remember back this multi-modular polyketide synthase. That the backbone is made by assembling these 2 and 3 carbon units, and then essentially cyclizing them. But then there are a whole host of other enzymes that are called tailoring enzymes, and those generally then they can methylate, oxidize to different states the intact molecule. So that's how you get that's how the organism makes these libraries of compounds. Generally, you will find a suite of compounds when you find a structural type that the organism is making. So they do that by these tailoring enzymes. Mm-hmm. It's also true in the case of proteins that there are some glycosylation motifs as well for proteins that are, of course, critical to their biological function. They also turn out to be critical in some sense to natural products, small molecules binding them. There are distinct cases where the sugars play an important role in that. So it goes both ways.
Mm-hmm. So that complicates the drug discovery, I guess, of natural products in some ways. Or do you just want to avoid natural products that are post-translationally modified?
Actually, if you're talking about a drug, then you're talking about just one molecule, right? So what we do then is let's say we find a hit that's a distinct molecule, and it has a suite of brothers and sisters. We will then, the bioprocess, when you finally make the drug, first you find out which one of those, or some analog of it, is the best one, has the best properties, safety, etc. activity, you then, if you're going to make it and we do it by fermentation, let's say, you will engineer the strain, first of all, to make only that one, right? So we'll modify the genes, take out the genes that make all the other brothers and sisters, and also you also have to modify the strain to make a lot of it, for it to be commercially viable. Some of these molecules are made in very, very trace amounts. So there's ways to get around that.
And then my last question is, it seems like, you could sort of say biologicals are natural products in some ways. And, I just wondered if, we don't actually have a session on biologicals, but I wondered if it would be possible for you or any of the others to give your thoughts about biologicals in terms of, we mentioned that there aren't that many genome proteins that become drugs, right? But these are natural products, and I guess they're larger molecules, they're harder to engineer and so on. But at the same time, there's so much interest now in biologicals as drugs. It just seems maybe we can learn something in the crossover here, and I'm not sure what it is.
Yeah, there's certainly with antibodies or other or therapeutic proteins, they are the endogenous drug typically, right? You're generally using the protein that's made already, right, and using it therapeutically. But what you find is that they have terrific specificity, right? They only bind their target, right? But they don't oftentimes have much potency, and so that's a drawback to biologics. In some cases they are, right? They're fine, but other times the potency is not there. So what you can actually do, and this is now being done recently. There's just a drug launched earlier this year called Etcetras. It's an antibody-drug conjugate. It has a monoclonal antibody to which is linked a very, very potent cytotoxin, right? So, so the antibody provides the antigen specificity. It goes to the tumor cell. It's raised up against a specific protein at the tumor cell surface. When it gets there, the antibody gets brought inside, internalized, and then the natural product is released. That natural product is a very, very cytotoxic molecule. You could never administer it systemically, but the antibody brings it, to the site of action.
Yeah, and with the endogenous natural products, like I said, at very low levels, the problem is when you change the pharmaceuticals, go back and discover all target pharmacology.
Yeah, natural products have the same safety issues and concerns and efficacy issues that quote, small molecules do. You still need to build in the precise attributes.
I'm sorry, I don't want to monopolize. I just have, if I might ask one more question. Is there any possibility that, especially with the trophic factors, that natural products that might be a source of mimetics for some of the trophic factors that are biologicals?
It's a little out of my area, but I don't see why not. There are these beyond rule of 5 molecules. They mimic protein motifs, right? So alpha helices, those things that are involved in protein-protein interactions.
Morning. I've been hearing a lot about having mechanism of action present in your druggability. But for example, is that a suggestion, a rule, a mandate? Because a lot of drugs we've used without knowing the mechanism of action. And sometimes the mechanism of action that is proposed, for example, for some compounds, doesn't make it. It really, when you look at it in detail, it doesn't make any sense.
Well, there's also a problem with terminology. You talk about druggability. There's the chemist's definition, and there's the pharmacologist's definition. You have the same thing with mechanism of action, the biological mechanism of the pathway that you're looking at. Especially true for phenotypic screening, where you don't have a molecular target, and it's not a mandate that those things, or there are drugs that are approved and in clinical use, and there are ones that are currently now in clinical trials. We don't know the exact molecular target by which you get the efficacy, but in fact, you know that the mechanism by which they do it, which is to modulate the CNS, they might modulate a neurotoxic molecule in foods. So that would be a biological mechanism, but not a molecular mechanism.
I can comment. It's not an absolute necessity, but it's a great asset because it's very difficult to convince people to take a molecule forward into the clinic if you really don't have a distinct idea of how it works or how it should work if it does work eventually.
Well, but look at Keppra. Look at Keppra. Keppra is an anticonvulsant, which we maybe know something about the mechanism of action map right now.
Yeah.
And those are about the recent ones.
Yeah, your point is a very good one, particularly with older drugs. What we thought was the mechanism, it doesn't turn out to be. Yeah.
Well, for Lyrica, for example,
Yeah.
the actual molecular target was not the one that worked in the low doses to get to the compound the drug was developed. You got the CNS that had an effect in certain models, but when you got to the dosing and linking the target engagement to the efficacy, it was not the target which was developed. But so you're getting questions that have to do with financial decisions, biases by investors, going up the food chain of companies and review panels versus what's required for an FDA package that they can get into exactly make it tomorrow. So that's good one to keep a hold of.
Hi, I hope this will engage the whole panel. We heard in the last talk that you were addressing a CNS audience, and then you made provocative comments about undruggable targets and outside of the rule of five and whatnot. So we have a lot of difficulty developing large molecules in CNS areas. So, but we really need natural products right now. And you mentioned nicotine. And there are a lot of low molecular weight alkaloidal compounds out there in plants, probably not in your fermentations. But as a CNS audience, and I hope the other speakers will plead along with me, we don't need to study FK506. We don't need any more anti-infectives. We need low molecular weight compounds that get through membranes, get to where they're supposed to go. And now we have such great analytical tools and spectroscopic tools that we can identify these things pretty quickly from mixtures. So is there going to be this turnabout where, why don't we find more nicotine, caffeine, cocaine, morphine? I mean, plants give these low molecular weight alkaloids up all the time, but we never hear about them from natural products chemists.
I'm trying to get my colleagues here involved. So plants are a whole different aspect of small molecule chemistry. Yes, you're absolutely right. They make vast numbers of very interesting pharmacologically active compounds. But they're a far different thing to advance. Generally, you will do more synthesis with plant-based leads than you will with microbial-based leads, because you can't. Eventually, you can make plant compounds in large amounts. Either grow it in a field or ferment it in plant cell culture. But to get there, you have to make a very large investment. So generally, you do a lot more synthesis up front, and the way you find the molecules is a little more challenging, because actually the plant, the dynamic range between plant compounds is huge. So I can tell you I agree with your premise that the chemistry of plants needs to be explored in a deeper way, particularly with the means we have now. And there are some small companies that are doing that. And there are companies that are. I know Novartis has a sizable natural products plant effort that they're looking at Chinese medicinal plants, compounds they're from. So
There's an
yeah.
area called nutraceuticals, from which plant-derived materials now pursue
Yeah.
now the purification scale of the system.
Yeah, it's a different paradigm. I think plant compounds are wonderful. It's great stuff there. It's just a different process. We don't do it just because you can only do so many things.
And good morning. My name is Marek Brzezinski from UCSF, and thank you much for a wonderful session. One line stuck with me is what you said: kill early and fast. And because it touched on a dilemma that I have when I review papers on Alzheimer, or when I think about studies in the future, because Alzheimer is a little bit different than other diseases. Like if I deal with hypotension, if I deal with shortness of breath, it is very quick to do. I see change immediately. So it is easy to see if the drug works or if the drug doesn't work. With Alzheimer, we see a progressive disease over years. So I sometimes, will be very simple-minded to think that you will see a change immediately. So the question for you that I have, how long do you actually personally wait to see effect? What actually is the outcome that you're looking for in your animal studies if you look for phenotypical? And do you think that potentially the delay in discovery of drugs is because we are too, we don't have patience with drugs and we don't follow up for a long time, and we just decide that drugs are negative because we didn't follow up for a long time? And if I'm planning to review studies, clinical studies, or planning in the future to do some, how long do I have to wait, or how much time should we give a drug? 1 year, 2 years, 3 years before we decide that something doesn't work?
Well, you're getting into the later stage. The discussion was mostly about the early stage. The idea was how to quickly get to something that you're going to have reduced risk when you actually go to the animal studies and do longer term. Because in fact, Alzheimer's disease, generally you're going to, even if you do a short animal screen, you eventually might want to do once a year, which process is clear laid out. But the key thing is to get to a discrete number of things that are no-goes. So there's an estimate as high as 98% of things that do not have sufficient, permeation in the brain. So the idea of the early discovery is kill those early if you're going to go for Alzheimer's. You probably want something for oral bioavailability. You don't want to have something patients come in and get ICD injection. So you're going to, so the molecular properties are there. So that's the idea is early on, make things that allow you to do the preclinical and clinical studies with high value compounds is the concept of this session. There'll be other ones, people behind you will not maybe talk about clinical trials too.
I have a general question about what you mean by brain accumulation or BBB permeability. And the question is that the brain is a bit more complicated than just the brain. So the question is, does one ever look for regional specificity? Does one ever look at deep matter structures versus cortex? Does one ever compare spinal cord to brain? And for the MS researchers, do you have a concern about white matter accumulation versus gray matter? And are these rules of 5, are they pertinent? Do they change if you're considering like gray matter versus white matter? A lot of questions about it.
Sure, I'll try to take them one at a time here, but essentially, people have done particular studies where they've looked at diffusion into specific regions or locales within the CNS. I think I was speaking more from a general terminology where it's partially context specific, so depending on what you're working with is going to influence what parts of the CNS that you're going to need penetration into. And again, it's a bit of an equilibrium or a kinetic argument as well, because it's going to be relevant to the therapeutic window in which you're applying it. And to Marty's point, where you showed that graph, if you're thinking about, let's say, a stroke scenario, you're going to have a specific window in which you're going to be treating. And then if you're affecting a disease-modifying target, like a biosynthetic process, you're only going to need that penetration to occur at a sufficient level for a minute time, and then it can disperse, go away, be metabolized, efflux out. But then if you're going for a palliative treatment, maybe you do need to escalate the penetration over a broad range at a larger concentration. So a bit of a, I'm sure it's a catch, but.
Well, like in regards to Alzheimer's disease, if we want to, one possibility might be to target the hippocampus. So if you're doing drug discovery, would you, I mean, I'm fairly new to this field, would you then sacrifice your mice and look at brain accumulation in the hippocampus? Is that a requirement?
Having done the studies before, you would actually have to dose pretty large and very sensitive analytical techniques, I think, to sufficiently detect the drug in that region. Just having worked with the mouse PK myself, it's easier to use it as a quick and dirty yes/no answer, I think is the best way to apply it. If you're actually going to screen in your lab to say, yes, the drug is getting in the brain or not, it's easier just to do a whole homogenization on the brain and say, yes, I see drug there, and this is the estimated concentration relative to the plasma. And then you can do more detailed PK studies later on. And just thinking about the discovery engine, if you have 5 to 10 candidates and you're going to think about going to an efficacy model, this is just a quick way to figure out, okay, compounds 1 and 3 are 3 times higher in the brain than the plasma, but 4 through 10 don't get into the brain significantly. So it's more of a quick, dirty response, I guess, is the easiest way to use it.
If I could follow up on two, remind me about dosing, because widely distributed targets work. Approved drugs that go to widely distributed targets. Generally in the CNS, when you're looking at these sort of rule of 4 type rule of thumb, things that are developed that way generally diffuse throughout the CNS fairly well in case of these studies. So in fact, with larger animals, where you look either at regional MRI spectroscopy, or you actually do biopsies and look at those with highly sensitive techniques, they pretty much diffuse there. So really the question that's more relevant based on history and precedent is dosing, and that has to do with widely distributed one or something that's regionally localized. Is that where your change is occurring? Because you're basically looking to restore homeostasis. So in this would be for early stage AD or progression versus late stage. This has to do again with the time window. You're looking your animal model and your dosing paradigms to wait until you have late stage pulmonary disease. You have to reverse it. That's one thing. Versus looking for things that are correlated with trends or that mechanistically contribute to synaptic dysfunction, and you want to go after those. And until we get them, we don't know. But whether the drug permeates into the area and has good diffusion parameters in your pulmonary, basically we want that as a first step requirement, but how that's like efficacy is has to be.
I'd like to talk about the concept of the importance of mechanism of action briefly. I think we need to consider all of this as a multidimensional paradigm. Regulatory approval doesn't require mechanism of action. Regulatory approval requires efficacy and safety. We tend to think very dichotomously. We have a drug, we have a target, we show the target is engaged, it changes a biomarker, so we have identified the mechanism of action. But the reality is there is no drug that acts at only one target. It is distributed throughout the body. There may be targets that it acts on in other tissues at different potencies, and over a period of time these can accumulate. We can think about the post-marketing failures of Vioxx. We can think about the problems of developing glitazones. There's systems pharmacology that I'll be addressing a bit later today that needs to be taken into account. So knowledge of mechanism of action is a de-risking factor. And it is very important in being able to tease out signals that may become apparent in a clinical trial, and then certainly in post-marketing use of the drug once it's been approved by the regulators. So it's not required up front necessarily. It helps. It's not required in early stages of drug discovery, but I think we need to realize that the reason that we're doing this is because we want to treat patients. And I think we need to take it upon ourselves to do everything we can to understand mechanism of action and make that a priority. And not only mechanism of action, but mechanisms, the impacts of these drugs on homeostatic systems. We see these complex diagrams with vectors going off in all different sorts of network pathways. We need to understand that because that's what we're impacting on when we're treating patients.
That's a good point. It's risk reduction. Along those lines of theory, it's a lot of times discussions with this. It's always good to think back. Richard Viagra, Vioxx, but you have non-steroidal NSAIDs, and you might have a target. That target activity may increase after injury or stress, and it may increase across multiple tissues.
best example of this over the past year is the story of semagacistat, where there was a pristine mechanism of action, but the system pharmacology was not taken into account.
Yeah, your points are right on that. The point I think is important to consider as well is that when you, and I'm not a clinical scientist, but when you go into the clinic, you're looking for two things. Hopefully your drug's going to work, and more than likely it won't, at least in proof of concept. But when it doesn't work, then you have some idea of why it doesn't work, or that at least your concept is not a proven concept. So you do need to know the target if you're going to design a proof of concept that's going to tell you something. And that we have learned painfully, I think. The other thing has to do with precision medicine, right? So if you're going to target subpopulations of patients, I think it's also important to have a pretty good understanding if you're going to learn what subpopulations you're going to go after, because isn't that one of the problems we have? We're only getting a few patients out of a disease group where it works.
And that may not give you a similar background in the clinical trial, but there
Yeah.
could be very important subpopulation retrospective analyses. The other point is we were talking earlier about the iterative process between physical chemical properties, pharmacodynamics, that sort of process that goes into a drug discovery program. There also needs to be this iterative process between discovery research and clinical research. By understanding mechanisms of action and overlapping networks, you may get a failed clinical trial, but get kernels, gold nuggets out of that trial that can then be handed back to the discovery scientists to refine the networks that they're impacting on with relatives of their chemical series.
So, comment on Barry's comment and then my original comment. Getting your foot in the door with big pharma is very difficult without a mechanism of action and a single protein target. So regardless about FDA and everything else and the way the world should be, if you don't have a single protein target and a mechanism, it's very difficult. So my original comment was going to be associated, I missed your talk, Marty, I know myth was in the title, a comment about the numbers of compounds that need to be made in any particular project. Going from compound 30 to compound 130, I would submit that there's rather an incremental change in the properties of the molecule. And since in general one is not going to look at the bioavailability or the toxicity of compounds 30 to 130, in a pharmaceutical company one might have the resources to make 100, 200 compounds. But in an academic environment, if one's making a smart, focused library of compounds, does one really need to go above 30 or 40 compounds?
No magic number.
So we can include that as one of Marty's myths then. The fact that you have to synthesize hundreds of compounds to get a lead and a backup.
Yeah, where the state of the art is right now is that there actually are fewer higher value compounds being made given the process.
And that has to do with the platforms, the computational platforms that are now available. They're for tox, safety, properties like permeability. Some of the models are pretty good now. So you don't need to make those molecules just to find out they're not permeable. It used to be, and I'm not a medicinal chemist by training, but it used to be a badge of honor in big pharma. We made 1,200 compounds to get this final clinical candidate. The opposite is now true. No one wants to hear that you made 1,200 compounds. They want to hear that you made 20 compounds, right, and you have your clinical candidate. So it is different now. Sometimes you still need to do it.
Thanks very much.
You mentioned